Gregorio Iraola, who focus on the importance of the human microbiome. So Gregorio, the floor is yours. Thank you.
Okay. Are you seeing now?
Yes, perfectly. Thank you.
Okay, great. So I saw that I was presented as founder of Kings Bio, that is my startup company. But in the company we work with phages and applications of phages in different problems related with bacteria that can be harmful for human and animal health. But today, what I wanted to share with you is some progress on an academic project that I am also leading in the context of a collaboration with the different Latin American and European countries that is focused on studying the human microbiome of underrepresented populations. This is in the context of my position as principal investigator at the Institute Pasteur in Montevideo in Uruguay, at the Microbial Genomics Lab. So, a very brief heads up on the human microbiome. We will be focused on the gut microbiome in particular today, that is basically dominated by bacteria, but also we will see that there is a very important fraction of archaea, fungi, and viruses in particular, and phages in particular. Most of these bags in the gut microbiome are commensals, mutualists, but also can be potential pathogens for us. And in the recent years, we have been discovering that different diseases related to the gastrointestinal tract or not are consequence or are caused by different dysbiotic processes occurring in this gut microbiota. This dysbiosis is defined as deviations of the normal distribution and composition of bacteria. But another thing that is important is that we have been discovered that the human gut microbiome genetic and taxonomic diversity is yet unsaturated. What this mean? Well, this means that every new region or population that we analyze uncovers new species that were previously unseen and new genetic diversity that was previously unknown. And this is particularly important because today most human microbiome studies have been conducted in high-income countries, particularly from the US, Western Europe, and China. So this map shows the distribution of public data sets from human gut microbiome. As you can see, there are entire regions in the world, including Latin America, Africa, Eastern Europe, Middle East, Southeast Asia, that today are like black holes of information regarding the human microbiome. And this deserves to be studied to get a better picture of what's going on with the microbiome at the regional level, at the local level, and how this can impact our health. So we focused, because I am based in Latin America, in Uruguay, we started with uncovering what is going on in Latin America. Latin America is a very diverse continent with most of the countries are low and middle income countries. We have had different immigration waves. We still have in many countries indigenous communities that lives in a more native lifestyle. We have a mixture of cultures and ways of living that is very, very complex. So we expected as well a very, very complex picture of the human gut microbiome. Also, we have at the continental level high incidence of some inflammation-driven diseases like obesity or diabetes or different type of cancers that can be potentially linked to the human gut microbiome. So to start, we built what I call the Latin Biota Consortium that today is composed by more than 10 countries, but the results I'm going to show you today are based on these 8 countries that initially submitted samples for performing metagenomic sequencing of the human microbiome. So these countries are Mexico, Ecuador, Colombia, Brazil, Uruguay, Bolivia, Chile, and Argentina. So we build this network of collaboration that is ongoing and is growing. We will see at the end. But basically, from 2019 to 2021, we ran the phase 1 of the consortium that was focused on studying the microbiome of healthy adults in the continent. So to do this, we achieved to collect over 600 samples, fecal samples from 600 people in these 8 countries. And we apply a shotgun metagenomics approach over all these samples. We were able to build over 18,000 mags. We have also samples from urban and rural lifestyles, not only rural native lifestyles, not only urban lifestyle, sorry. So we have a quite comprehensive representation of the different way of living in the continent. And we gathered some metadata from these people. So we started to characterize the microbiome in the region. Basically, when we started to analyze the mags, we discovered that interesting proportion of them did not match with previous catalogs of bacterial genomes recovered from the rest of the world. And at the end, we uncovered that over 300 species, bacterial species, were novel in the Latin Biota dataset in comparison with all these global public dataset that we built from public repositories that was over 30,000 gut metagenomes from different parts of the world. So with these over 300 newly described gut bacterial species, we built a phylogeny. We discovered that these species were basically widespread across the phylogeny, and we started to measure how was the prevalence of these newly described species in Latin America, in the rest of the continents. And surprisingly, we discovered that most of these species were highly prevalent in Africa. So we detected this Latin America-Africa connection in terms of sharing of this particular newly described bacterial species. And when we analyze which taxa were most represented in these new species, we detected the Oscillospirales order as one of the top represented taxa within these new species. And when we analyzed the prevalence of these Oscillospirales taxon in different regions of the world, we detected that most of these new Oscillospirales species follow a vanish-like pattern. A vanish-like pattern is defined as species that tend to be very prevalent in less industrialized native or rural lifestyles and tend to disappear in the gut microbiomes of populations living in highly industrialized lifestyles or settings. So with this, we discover this new set of species and taxa that are potentially associated with less industrialized populations. This can have this can be related with the diet, with the way of in the way of living in general, the culture, well, different aspects that have been associated with this. But the interesting thing is we have this new set of species that are now available to be studied more in depth. And indeed, we focused then on the Oscillospirales group, and we built basically we did a population structure analysis, and we detected some Oscillospirales lineages that are particularly enriched in Latin American populations with respect to the rest of the world. And once we did this, we then perform a functional analysis of these genomes, these Oscillospirales genomes, to detect which functions could be more enriched in Oscillospirales that are very prevalent in Latin America with respect to the rest of the world. And one of the things that caught our attention was that among the defense mechanisms coded in the genomes of these Oscillospirales that are prevalent in Latin America, most of these were AMR genes. So this, of course, motivated us to perform a much more comprehensive AMR analysis at the whole set of taxa that were recovered from Latin biota and from the rest of the world. And once we got the results, we were very surprised because basically we detected that Latin America was the region in the world with the highest prevalence of AMR genes in the gut microbiome of healthy adults. So this, of course, from different technical aspects of the data analysis, don't consider genes that are being carried on plasmids, because the mag reconstruction leave the plasmid out. So this means that all these genes that we are detecting are probably coded in the bacterial chromosomes of commensal bacteria, meaning that healthy adults in Latin America in particular, but in different parts of the world, have a huge reservoir of potential AMR genes that are coded in the bacterial chromosomes, but can have the potential in the future to shamp to any horizontal gene transfer platform like plasmids or even phages or even other platforms, and constitute future risk for human health and for the AMR pandemic. And also, we not only focuses on the bacteria, but also reconstruct viral species, and particular most of these viral species are phage species that infect bacteria. And in comparison with what is known from other databases, we uncover over 9,000 new viral species that correlate in their diversity with the bacterial diversity in the general datasets. This makes sense because the viruses infect the bacteria, and they are an ecological balance there. But was most interesting for us that when we try to assign the host, the bacterial host, to this new described viral species, we found that the Oscillospiraceae family was one of the top families being infected by this new viral species that we detected. So returning back to what we saw from the bacteria and the importance of the Oscillospirales group, we are now studying how this or what is going on here, because we have a important set of viruses that are also infecting these Oscillospirales, and apparently in Latin America, for any reason, these Oscillospirales are important because are much more prevalent than in the rest of the world. So there should be an equilibrium in any way, a biological equilibrium in terms of which virus can infect and kill and control in any way the oscillospirales that we are detecting and we are identifying as important for the normal Latin American microbiome. So this is we are almost finishing. With all these sets of bacterial and viral genomes, we also studied the functional repertoire of 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 molecular and biological functions coded in all the genes we could recover from this. And interestingly, we detected over 200,000 new protein clusters that are less than 90% identical to any other protein known before. And out of these 200,000, around 30,000, we couldn't annotate any function, any noun function. So we have over 30,000 new proteins that are totally unknown for us. We don't know which function they can be playing. So it's some novel biology in there, and of course different potential biotech applications through the characterization of these proteins. So to conclude, we have run the phase 1 of Latin Biota, analyzing over 600 samples. This contributed to the discovery of over 300 new bacterial species and over 9,000 new viral species. Also, we contributed to enhance the quality of previously known gut reference genomes because we have some mags from species that were previously detected in other regions, but now we have better quality representative genomes of these species. As I mentioned, novel bacterial species are mostly related with the Oscillospirales order. I didn't mention it, but we also developed a new method for high accuracy recovery of viral species from the gut metagenomes, and this also allow us to detect for first time in Latin America some particular lineages of some phages that were thought to be absent in some regions of the world, and now we are improving the characterization. Also, I didn't put in the result for a matter of time that we discovered that the virome is much more informative to predict the origin of a sample, the geographic origin of a sample, than the bacterial part of the microbiome. So we have some machine learning developed there to predict the geographical origin of a sample from microbiome data. And as I mentioned at the end, Latin Biota contains over 200,000 new protein clusters that need to be studied more in depth to understand their function and their biological role. And the Latin American microbiomes have the highest prevalence of IMR genes as we saw previously. What is next? Well, I have built this network. We have recently got some funding, some extra funding to expand the concept we developed with the Latin Biota toward other underrepresented populations. And we are doing this through the Pasteur network, which involves different institutes in the world, and the red ones that you see are now involved in sampling and sequencing metagenomes from population from these countries, basically Latin America, Northern Africa, and Eastern Europe. And this is going to constitute the phase 2 of Latin Biota. We expect to reach over 3,000 samples and expand new geographic areas, lifestyles, and of course, welcoming new collaborators. We also expect to explore other body sites, not only the gut microbiome, but we are particularly interested in the oral microbiome. And we also plan to integrate other sequencing technologies, not only short read Illumina as we did in this initial phase, but we also want to include long reads and some high-C analysis to improve the quality and the resolution, in particular for bacterial phage and bacterial plasmid relationships that we can uncover using these technologies. And in the midterm, we also want to start focusing on some other categories and some other cohorts that could be, for example, diseases that are important for different regions we are studying, because now we have a very good baseline of what is normal in Latin America, at least, that was previously unknown. And we have this starting point to perform much more comprehensive comparisons with cohorts of different diseases that can affect or be affected by changes in the gut microbiome. So that's the end. Thank you very much for the opportunity of sharing this project and my results. These are my contacts. Any case or any question you have, please reach out, and I will be happy to discuss with you about this. Thank you very much.
Thank you, Gregorio, for the contribution, and for our audience, just a quick heads up that this is not the end of this topic and our collaborative work with Gregorio, because I'm sure in the next year we'll continue this conversation more in regards in the antimicrobial resistance. So stay tuned, and thank you again, Gregorio.